We are blessed to have several leading creation speakers for tonight's important event. Our speakers include Dr. Randy Guliuzza, Dr. Brian Thomas, Dr. Dan Biddle, and Dr. Carl Werner. Get ready for 80 minutes of the best lines of evidence supporting young earth creation.
Dr. Brian Thomas received his master's in biotechnology in 1999 from Stephen F. Austin State University in Texas and a PhD in paleobiochemistry in 2019 from the University of Liverpool. He taught at a Christian junior high and high schools in Texas and taught biology, chemistry, anatomy, and is an adjunct assistant professor at Dallas area universities. In 2008, Dr. Thomas joined the Institute for Creation Research as a science writer, contributing news and magazine articles speaking on creation issues, and is researching original tissue fossils. He was appointed as a research associate in 2019 and a research scientist in 2021. Dr. Thomas, thank you for your time tonight. How are you?
I'm doing great. Thank you, Sam and Donnie. So am I up and running? Can everyone see my opening slide says fossil proteins support
Dr.
scripture?
Thomas, the presentation is up and you're good to go.
Okay, thank you very much. Thank you for joining in, everybody. And I just got to tell you, I did not believe the flood, at all. I didn't believe the Bible when I was growing up. And the first person I met who actually believed the flood, he was a different guy. And funny enough, I was on the phone with him earlier. He happened to call me. This is 35 years later. And he said, he said, "What are you doing tonight?" I said, "Well, I've got this, this interview on the YouTube or whatever it's going to be on." And he said, "Oh, my wife and I are signed up for that." So he may be listening to me right now. So Pastor Kurt Wise of Alvin Bible Church, if you're out there, man, I just so appreciate you and love you, because you challenged me when I was a young Christian, and I believed in evolution, but I also knew Jesus was real. But I was like, what's this about the flood? I mean, everyone scoffed at the flood. Like, why is the flood even described in the Bible? I think it's there for us to just, poke fun at. And he said, well, what if the flood really happened? And I just thought, no one's this is blowing my mind even to think like this. What if the flood really happened? What would you expect to see? And what if the flood actually caused all these rock layers that we have and the fossils in them, and maybe the flood is why we even have fossils. And after all, we don't have fossils forming today. And I thought, oh, well, that's crazy. Why would you think that? But then he kept asking me. And so I started to investigate, and I thought, what would I expect if the flood really happened? And so we're going to look at some expectations first, and then I would like to lay out the idea that every Christian should feel confident that that the Bible got it right about not just the fact that the flood happened, but when the flood happened. So I'd like to present for the next 15 minutes some evidence for the timing that the Bible gives for Noah's flood at about 4,500 years ago. Is there evidence in the rocks, in the fossils, that fits, generally speaking, with this recent timing, recent as compared to what I was taught, which is that these rock layers go for tens of millions of years and the lowest ones are hundreds of millions of years? This is a very different way to think about history. So what would we expect about to see in the fossils and the rock layers if they were indeed deposited during Noah's 1 year long flood, according to the scripture? So let's look at what Peter said in the New Testament. Looks like to me he took the Old Testament very straightforwardly, because he says this: "For this they," and he had just been talking about the scoffers who willfully forget. What? That by the word of God, that is by the supernatural, the heavens were. So the heavens came into being from the supernatural of old, and the earth. So the heavens and the earth. He's talking about Genesis 1:1. In the beginning, God created the heavens and the earth. Well, I was taught in the beginning, nature brought forth the heavens and the earth. And this is saying a completely different thing. Then he says this, standing out of the water, that earth, standing out of water and in the water, by which the world that then existed, which is to say the pre-flood world, perished. Perished. Garden of Eden, perished. The continents, maybe, perished. Whatever was the hills, perished. The oceans back then perished, being flooded with water. He says the world. And so, did he mean the whole world? And if so, what would we expect to see? Well, we've developed a model over the last 50 plus years of ministry at the Institute for Creation Research, where I work in Dallas. We had 36 years or so in San Diego area. Now we've been in Dallas since 2007, and we're on a second or so generation of scientists researching the scientific evidence that supports Scripture. And when my friend Kurt, Pastor Kurt, challenged me on this, and I started to investigate, I read an ICR. That's where I work now. I read an ICR book with the intent to demolish it and to scoff at it and poke fun at it. But by the time I finished reading it, I thought, this explains the world I live in better than anything I had ever seen. So our geologists, especially Dr. Clary, have mapped the estimated sea level based on the thicknesses of the rock layers. So you have to have a lot of water in order to deposit a lot of sediment, high volume of water, high volume of sediment. And so we've mapped that onto the days of the flood. And so instead of representing hundreds of thousands or tens of millions or hundreds of millions of years, we're looking at this whole entire stack of rocks. And on the left, you see, the familiar rock systems, Cambrian, Ordovician, et cetera, from the bottom up. But on the right, we have these systems of rock packages where the bottom of each one is a sandstone, and then the sediment gets finer and smaller up to limestone and then mudstone at the top in each of these packages. These are called mega sequences, and we actually see these on the Earth. So this is a map in the center here showing the height of each of these rock packages, where we find them on the Earth. And we can imagine during this flood, it wasn't just 40 days and 40 nights. I ask audiences across the world where I've spoken, how long was the flood? And anyone with any familiarity says 40 days and 40 nights, right? That was just the start. The scripture says that after day 150, that's when all the high hills of that pre-flood world were completely covered. So we have a photo here of the Japanese tsunami that struck. I mean, there's nothing that can withstand the force of moving water, especially when it combines with mud, and it turns into a juggernaut, literally, and it just blasts through whatever. So imagine this is just a teeny tiny modern day tsunami. Imagine one where the water level is 500 feet, 1,000 feet high, and whatever is scraping across the earth is going to get absolutely bulldozed. So our research here, based on thousands, tens of thousands of columns of rock, boreholes that have been drilled, have mapped out the extent of continuous rock layers. Look at this. First layers of the flood, just in those areas, later in the flood, and then finally with the Tejas mega sequence, the last layer that the flood deposited is offshore, and it's worldwide. And so we would expect that, because it says in the scripture that all the water came off the surface of the earth, and there it is sliding off the continents at the end of the world, at the end of the flood there. And this is a map developed, again, as I say, from about 10 years of diligent research at the Institute. So these are some of the kinds of things we'd expect. What we're saying is this, in essence, all the rock layers, the whole stack, well, not all literally, but virtually all, these rock layers were deposited in just 1 year. It sounds crazy. It sounded crazy to me when I first heard it, but But when you see the evidence that I study, which is proteins that are still in fossils, then it really starts to add up. Because if you see proteins in fossils in the Tejas megasequence, that's tens of millions of years old. You might think maybe proteins can last that long. But if you see them in the Zuni, or even further down in the Absarica, the Kaskaskia, and even down into the Sauk, which is, we typically call Cambrian. And yes, we found proteins in fossils still in the Cambrian, and it's designated as 500 million years old or more. And I'm thinking, how can a protein last that long? So that's the kind of presentation that we have. But we've been able to map out what to expect based on Genesis, and we can almost like pin signs to the rock layers as where we see them on the earth. Genesis 7, and the rain was upon the earth. So day 1, we have the Sauk sequence. And then by day 40, the waters increased and bear up the ark. And the waters prevailed and were increased greatly. Genesis 7:18, that's day 40. And then by day 150, the waters prevailed exceedingly. All the mountains were covered. The high hills, those are the high hills of the pre-flood world. And then after day 150, we have this the waters go off the surface of the earth there. So that's what to expect. What do we expect? We're expecting that the whole rock stack virtually was deposited in just 1 year. I know it sounds crazy, but when you see the evidence that we're uncovering with these fossils, it actually fits, and it makes me more confident than ever that if I'm a person who has been believing in the Bible, I've actually been believing not in spite of evidence, but in light of evidence. One of those evidences is the evidence of decay. And I mean biochemical decay. Like what is tissue made of? If we talk about skin or blood vessels or body tissues, they're made of proteins mostly. The proteins are the structural elements that hold body pieces together, and a lot of that. The long-lasting one is collagen. So I study collagen a lot. Here's a molecule of collagen, a model of it. Each of these little spheres in the top shows a single atom that's involved in combining together to make the amino acids. And the amino acids are linked together in a specific order in order to make a spiral. 3 of those spirals combine to make a fibril of collagen. But this collagen, tough as it is, it still falls apart because everything falls apart. Um, that's why we have to comb our hair in the mornings. The second law of thermodynamics says that all systems that have order lose organization over time. And certainly this happens. But the question is, how fast? How long does it take for collagen to turn into dust? Well, we investigated that. So here I am with my late and great colleague, Dr. Kevin Anderson, at a lab in Arizona, collaborating with him. And we have 3 water baths behind him. And these are some of the instruments and techniques that we use to test how long can collagen last. We take little pieces of bone, we put them in under heat because heat accelerates the decay, and we can use these lab bench measurements to estimate how long collagen can last. We published our results in a secular journal called Applied Spectroscopy. And here are our results. And I don't expect anyone to take anything from this except the fact that we do science. And basically, if you look at the second column, it shows colored images with lots of red at the top and then lots of green at the bottom. If you look at that column, the red is showing up collagen using a special microscope. There's lots of collagen when this bone starts. As you would expect, it starts with collagen. But as you go through time and under heat, with heat, it accelerates the decay of the protein. And the green business that's left over is not collagen. It's other parts of the bone. So we have all these samples that we've taken, and we basically showed that collagen doesn't last for 1,000,000 years. It doesn't last for 1,000,000 years. It can last for thousands of years under ideal circumstances, ideal conditions. But how long can it last? Not a million years. Uh, and so if you don't like my results, even though we've published them through secular peer review, then just go with these secular scientists. They don't believe the Bible. They have in fact, they're against it. And they published their results of a similar experiment. They said extrapolation from high temperature experimental decomposition rates using this activation energy suggests that at a constant temperature, it will take between 200,000 and 700,000 years at 10 degrees Celsius now for collagen to fall into dust, so fragments so small we can no longer detect them in an optimal burial environment. So both our results at the Institute for Creation Research and our colleagues and secular scientists agree that collagen cannot last a million years. That's the expectation. We don't expect proteins to last any more than a million years. So if we find proteins and they are in fossils, then we can say with some large degree of scientific certainty, based on repeatable lab bench decay experiments, that the collagen and therefore the proteins and therefore the rock layers that hold these fossils, they're not any older. They have to be younger than a million years. Well, which of the two worldviews? There's two worldviews here. One is, hundreds of millions of billions of years of nature doing everything, and the other worldview that I was introduced to um, and have come to now agree with is that God did it only thousands of years ago. So what do we find in the actual rocks? Well, speaking of collagen, here's a special microscope technique used to show the organized structuring of collagen that's in skin. So this is a Psittacosaurus dinosaur in China, in the Gobi Desert, and there it is with protein fibers still in the skin. Then there's the publication if anyone wants to look it up there. And I just have so many of these. In fact, I have 127 of these publications. Other scientists went and looked at this fossil turtle bone, and they said, this looks interesting. Let's analyze this bone. And they dissolved the mineral part of the bone away, which is called hydroxyapatite. They dissolved that away, it should leave nothing, because I was taught that minerals is all that's left when I was a kid, and textbooks and TV shows. Whenever you see a fossil, it's just minerals, minerals, minerals. All the original stuff is long gone. That can't last long enough for it to have persisted until today, tens of millions of years after the fact. I don't think it's a fact. I think because of these bone cells called osteocytes extracted from this bone, from this turtle fossil, not just dinosaurs, guys, it's all kinds of creatures that were fossilized. By the way, turtles are fossilized along with dinosaurs, as Dr. Werner will tell you, along with all kinds of other creatures, all in the same layers, including the birds that they were supposed to have evolved into. Cough, cough, cough. So what else do we have? We even have a recent report of DNA, or at least chromosomes, inside a fossil bird, buried in the same layers as dinosaurs. The bird's named Caudipteryx. This is a fun slide to read. Chromosomes in Caudipteryx chondrocytes. A chondrocyte is just a special kind of cell that manufactures cartilage. And so these cells are undergoing cell division, and they got frozen in sediment. And they extracted one of these cells, and they found right there some chromosome evidence. So the stain stuck right onto the DNA. And what else could it be sticking to except DNA, since it's a DNA-specific stain? And that was published there in Communications Biology. But this has been around for a while. Why haven't we been talking about it? Look at this, 1998. We found these are electron micrographs. So using an electron microscope, and on the top left, you see an image of a little tube coming out of a bone, and that tube is a blood vessel. And then image on the top right shows little bitty discs that are inside the blood vessel. And the author himself, he says these look like erythrocytes. What is that? Red blood cells still inside still inside the blood vessel where you'd expect them to be, and their iron content must have been present. Well, how are they still there today? So we have collagen, skin, we have blood vessels, we have red blood cells, which are made of hemoglobin, which is still holding the iron content that holds hemoglobin together. How do you get this to survive after all these eons? I don't think it's possible. The science shows it's not really possible. Well, here's another. By the way, that was the Gobi Desert, and now we're in Europe. We're looking at in Poland. These blood vessels from a Nothosaurus. Again, technically not a dinosaur, an extinct marine reptile, but blood vessels. And the authors of this paper said, 2016, well, there was a microscopic coating of mineral that stuck right onto the surface of the blood vessel and preserved them for tens of millions of years. That's a story. Did they test that story? The way to test it is what? Is to take blood vessels, coat them with mineral, and then put them through the decay rate experiment to see if they can last any longer than our experiments show that they can. And very recent results show that adding iron, this is an iron-based mineral that they're saying helps preserve these things. Adding iron to bone, these research, and I don't have the results in front of me, but you can look it up. Adding iron to bone actually accelerated the decay rate by bringing oxygen in, and it's a reactive oxygen, and it actually turns the protein to dust faster than it would otherwise. So anyway, it looks fresh. It looks young in Poland, and now here we are in North America with a hadrosaur. This one's called, it's not listed here, but I'll tell you, it's Brachylophosaurus canadensis from Canada. And we have blood vessels and bone cells on the bottom there. You can see with the branching dendrites and the blood vessels in the top center with little red discs there. What do those look like? Red blood cell looking things right there inside these blood vessel, well, they're blood vessels, and they sequence collagen. So this is incredible. This is actually, it is credible, but it's just amazing. And now this one of the last ones I'll show you, but there's so much more. This is a mosasaur, an extinct marine reptile, excavated from Kansas in 1967. I saw it on display in LA, where it's underneath a glass case. And I read this paper. These scientists said, look at this little red patch where its heart used to be. We think it's its heart. They said and they found the presence of hemoglobin. I mean, it's not perfect hemoglobin. It's really old hemoglobin, but there should be no hemoglobin if it's even 1 million years old, let alone enough hemoglobin for them to detect. And they said that we have the remains here, not only of the skin scales, which they did find, and the purple pigments of the retina in the eyeball of this creature there, but also residues of visceral organs. We're talking about the heart. So I saw it. I didn't know it was going to be there, but man, when I saw it, I was so excited. I said, somebody come take a picture of me. So this is a friend taking a picture of me, pointing to the remnants, the hemoglobin containing remnants of what might have been this creature's heart, and it's still red, and it's still there, and it's still on display. How many thousands of visitors to this museum in Los Angeles County have looked at this and thought, oh, what's interesting red stain, and never thought about it. And here it is. It's saying, I'm fresh, I'm young, I was not deposited that long ago. Well, these are the images that probably the best ones, and they were the first that got my attention anyways. In 2005 in the journal Science, we can see connective tissues that are labeled with black arrows on the bottom left, and they're tan colored. And we could see hemoglobin that they found, all from a Tyrannosaurus Rex leg bone, the femur bone. And so this was published, and they sequenced the collagen protein from this as well. So there's lots of evidence, and it keeps on mounting and mounting, and we keep compiling it. So this is a couple months old, but this is a list that we keep. There's the URL at the bottom, tinyurl.com. And if you guys want to keep abreast of all the literature, Every one of these lines, each line represents a technical, scientific, mainstream, peer-reviewed journal article that describes biomaterials, whether it's DNA or proteins or whole tissues in some cases, like blood vessel fragments. Wow. We list these on this web page. And actually, this says what we have 117. Well, guys, we've added 10 more since then. And the 127th one was our own. So I have a group of colleagues uh, that I have um, both within and outside creation circles. So I have evolutionary people that I work with because they're just interested in dinosaur bones, and they want to work with us on these projects. And we just got published in Analytical Chemistry. You could see we made the cover of the journal, and we found in an Edmontosaurus that Dr. Biddle actually donated, I'll tell you, to us for us to do this research. And he's about to present, so stay tuned for him. But we found and sequenced collagen protein in one of the hip bones of our Edmontosaurus. Spectacular result. So what's the end result here? The bottom line, we expect it to look fresh based on the timing that the Bible gives for the flood, thousands, not millions of years ago. We expect, based on protein decay, that protein shouldn't be there unless it's unless it was deposited thousands of years ago. And what do we find? We find exactly what's expected based on the Bible and based on the science of decay. We find proteins. Wow. So that brings us to the take-home question. The flood really happened. I mean, what does that mean for me? It means a lot of things. It means the Bible's right. It means, well, why did God judge the world? Was it because he was happy with everybody? No, he judged the world because of their sin, their wickedness. They only thought evil all the time, Genesis chapter 6. And so that means God will judge my sin, which means the whole reason we spend all this energy to deny the flood and to scoff at the flood like I used to do is not because there's no evidence for the flood. It really comes down to I don't want to admit that I'm a sinner and that I'm due to give an account on Judgment Day. I mean, this is a big deal, serious matter. But look at this. The same Bible that this evidence supports says that God sent his own son, the Lord Jesus, to save sinners, sinners just like me. And not, he knows we can't save ourselves. And he's just waiting for us to say, Lord, rescue me. And he is happy and thrilled to do it. So all you got to do is confess. God, you're right. I am a sinner. God, you're right. Your Bible is true. You judged the world once. Therefore, I'm confident that you will judge it again. I don't want to be a part of that judgment. Can you please do something for me? And then you learn about the Lord Jesus, that he died in my place, and then he rose again from the dead, so that I don't have to stay dead. I can rise up with him if I'm in him. And how am I in him? Just this: by confessing with my mouth that Jesus is Lord. This is from Romans chapter 10, and believing in my heart that God has raised him from the dead. So we see really solid science that supports the scripture in this way. I want to encourage our viewers to connect with ICR's YouTube page, and it's youtube.com/@ICR and we are populating our YouTube page with all kinds of great creation confirming videos, Bible confirming videos, and challenging. Some of them are challenging and some of them are fun. And I encourage you to do that. We also have a free magazine. It comes out every other month or so. It's called Acts and Facts. You can also sign up for that at icr.org. Thank you for the opportunity to share what I love to do, which is research proteins.
Wow, Dr. Thomas, that was an excellent presentation, and congratulations on the publication in Analytical Chemistry. Praise the Lord. That was great. Okay, well, we will bring in our next guest. Again, Dr. Thomas, appreciate that. Dr. G, Dr. Randy Guliuzza, it is great to have you here for our Science Confirms the Bible seminar. You are going to be giving a presentation Deconstructing the Engine of Evolution. Science shows that animals are pre-engineered to adapt and track their environments. I'm excited for this. I will let the audience know we will be wrapping up the night with an audience Q&A period. So if you hear anything said or presented during any of the talks tonight, feel free to send in your questions or challenges or objections, and we are going to have a Q&A period with the full panel as soon as we're done with the talks. And so, Dr. G, we're going to give you a formal introduction before we give you the floor for your presentation. Sam, would you do us the honors, brother?
I'd love to. So Dr. Randy Guliuzza is the president of the Institute for Creation Research. He is a leading biological design theorist and is actively expanding ICR's scientific research and heading the critical endeavor to develop a theory of biological design. He has represented ICR in several scientific debates at secular universities and other forums. He holds to a Doctor's of Medicine from the University of Minnesota and a Master's of Public Health from Harvard University. Dr. G also has a BS in Engineering from South Dakota School of Mines and Technology and a BA in Theology from the Moody Bible Institute. He was board certified in aerospace medicine, and he is a licensed professional engineer.
Dr. G, how are you tonight?
Oh, I'm doing great. Thank you so very much for this invitation to be here.
Well, it's our pleasure. I'm looking forward to this, and therefore I'm going to get your slides up here. I've got it up on the screen, and Dr. G, the floor is yours. Go ahead.
All right. Well, thank you very much. I hope everybody was listening very carefully to my colleague, Dr. Brian Thomas, there, with especially the gospel presentation that he had right at the end. Now, you probably already noticed immediately that I speak a lot faster than Dr. Thomas, and I really have to kind of move tonight because we have to cover a lot of material. In fact, even this title that I was assigned here is kind of long. So we need to move right back into what we're talking about. And everything I'm going to talk about tonight, almost everything, is already in this paper. So I know people will be watching this later on recording, so they can call up these slides later. But if you're interested in what I'm talking about, this was a paper that was published at Cedarville University at the ICC, International Conference on Creationism, just in 1923. So this will have a lot of the background information that we won't be able to cover in this short period here. But I do want to answer a challenge by some evolutionists, and there's a couple of them on the screen right there. And a lot of times we just push back against evolution, but they have this question a lot. What do you have to offer instead? You know, what do creationists have to offer instead, other than just saying God did it? And of course, we don't say that, but that's the expectation. In fact, an evolutionist gave a really good challenge that I think sets the stage for tonight's talk. He sent this in a text message, and he basically asked, here's my number one question for creationists, and in this case, he's including intelligent design people too. What's your research agenda? That is, what do you do next? And my biggest problem with this worldview, he's talking about ours, is simply that I've not heard of any positive consequences for research. What new research does your hypothesis help you do? And what would you do with a $5 million research grant? You know, that's a good question, and I hope to answer his challenge here tonight by saying that we actually have developed an alternative theory, a theory of biological design. And that's covered in the paper that I referenced at the very beginning, but I do want to cover some of the major points of a theory of biological design and really what a theory is going to help us do, what evolutionary theory helps people to do as well, is answer big questions in a particular field. And then after you do that, interpret what you see. And so evolutionary theory does that for evolutionists. So we need to develop a theory to answer the big questions as well and guide our interpretations. And the major observation in biology that really needs explanation is design. In other words, how do you account for the fact that creatures look so incredibly designed? And we see it, and evolutionists see it. It's the major question that needs to be answered, not only by their major organ systems and things, but also by their behavior, which is highly purposeful as well. And of course, how well they fit their environments, because, you know, birds look like they fit in the air and fish look like they fit in the ocean. How in the world do we explain all of that? Well, Dr. Paul Nelson, he came up with this wonderful, wonderful guide, which is that we need to begin doing biology as if Darwin had not been born. And I wish I would have said that myself, but he's basically calling for a fresh break, not to revise evolutionary theory in any way, but to come up with something completely different than Darwin's theory. And this is a quickie picture from Darwin's Field Book, and it really shows the beginning of a theory. And he begins up there with, I think. And a lot of times creationists and ID people criticize him for that. In other words, he's like thinking or speculating, but that's what a theory is. In many ways, it's a narrative to help guide our thinking. And you see there, he's speculating not just on the diversity of life, but why creatures look so incredibly designed, because every one of those nodes at the end is supposedly a creature that is so incredibly fit to their environment that they have actually developed exquisite design. So he begins by, I think, and that's what a theory is. A theory is a hypothesis. It's usually a working hypothesis that's supposedly a plausible explanation for how something works or how something originated. And Darwin came up with one of those theories, and it's really dominant these days. But what do we have to offer instead? We have a theory of biological design, and so I'm going to lay out some of my assumptions, the things that I think that are very, very contrary to evolutionary theory and specifically related to engineered biology. So I think that the reason why creatures look so incredibly engineered is because they are, in fact, engineered. And because of that, I also hypothesize that engineering principles will explain the operation of biological functions. I don't know of any evolutionists that would hypothesize that, but if I'm going with an engineering-based explanation, I would hypothesize that that's going to happen. In addition, a good theory makes predictions. So I predict under this theory that all biological systems can be reversed engineered. And evolutionists say that creationists don't make predictions, but this is one that would be easy to disprove. Just find one that can't be reverse engineered. In addition, I predict finding goal activity directed at every research level. That's from the organ system all the way down to the organs and the molecular level. I expect to find this activity at every single level. I also predict finding corresponding systems underlying similar engineered and biological functions. That's something that evolutionists wouldn't predict, but from an engineering perspective, I predict finding those as well. I predict finding identifiable control systems in organisms directing all of their operations. That's another thing that I would predict to find. And all of those predictions need to be set into the context of what I'm really going to talk about and what I'm not going to talk about. So in the realm of engineering, I would roll in all of the major biological functions of organisms, which is their growth and reproduction, adaptation, and metabolism. All organisms do those things. And so I am suggesting that in terms of how they operate, they all will be explained by engineering principles. But I am not talking about, specifically not talking about, life or consciousness or agency. And in fact, I don't know of anybody that has actually researched life directly. I don't know of a single scientific paper where anybody has held a beaker of life and done any experiments on life itself or consciousness for that matter. So I am not talking about those things. In fact, I would argue that they're not reducible to engineering principles or to biological functions, that they exist outside of them, that they're immaterial. And this is why humans really cannot create life, and creatures like you and me are not really machines. So I'm talking about the actual biological functions that organisms are doing: growth, reproduction, adaptation, and metabolism. And tonight we're going to look specifically at adaptation. So if we have everything in this context of a theory, where we have some hypotheses of what we're expecting to find and some predictions, then we need, as our first step, is to make new models. And so we want to put together a model of adaptation, an engineering-based model of adaptation that focuses on the organism. And because it's engineering-based, it eliminates all all references to mysticism. In other words, nothing that cannot be quantified and actually identified is put into our explanations whatsoever. If you can't quantify it and you can't identify it, we don't put it into a causal explanation. So let's look at a model that explains adaptation without any mysticism, focuses on the organism, and is engineering based. And that model that ICR has developed is called continuous environmental tracking. And other organizations have picked up on this model as well. And it also makes, like any good model, an hypothesis. And the hypothesis is that what creatures seem to be doing is continuously tracking environmental changes. And this is not that they're passive in the slightest, but they are active problem solvers. And they're actively tracking what's happening around them, all the way from the level of their metabolism and homeostasis up to acclimation and even multi-generational changes. And because they're able to purposely track, they're able to have purposeful adaptations. Now, this is very, very different from an evolutionary explanation. Everything is active by the organism. And so that's why we hypothesize that organisms are tracking. And how they're tracking is due to many innate systems that they're actually activating. And therefore, I expect the characteristics of what they're doing to be consistent with the characteristics of design systems. And so there are several distinguishing features of continuous environmental tracking compared to Darwinian selectionism. First, CET is organism focused. It's not focusing on the environment. Anything that any of those creatures you see on the screen can and ever will be able to do is inside of them. Any way they relate to each other or their environment is inside of them. So if you want to figure out how they operate, you focus on the organism, and that's where you'll find your explanations for adaptation. Second, as I've already alluded to, it's an engineering-based explanation. Therefore, from that, we make all of those predictions of what we expect to find and how we expect these organisms to operate and adapt. So one of the engineering-based expectations I have is that their capacity to adapt is built in upfront. So just as the engineers surveyed all the different environments that a space shuttle would need to go to and expected all of those challenges, then they built into that space shuttle the ability to solve those challenges. And therefore, from an engineering perspective, I expect the solutions to precede the challenges, and not from an evolutionary perspective as due to those challenges. So the ability to adapt to many different environments, of course, it's limited, just like any engineered thing. It has parameters that it operates within. But one major distinction is that those are built in up front. And therefore, I expect to find corresponding system elements. So on the left-hand side of the screen, people are looking at an autonomous car that can drive itself around challenges, around storms. And it does so because it has many built-in sensors, innate logic mechanisms, and a response capability to do those things. Therefore, on the right-hand side of the screen, when I'm looking at guinea pigs under this sign that says guinea pigs beat climate change by tweaking their own DNA, I'm expecting to find those same system elements of sensors, innate logic, and this response capacity. And in this case, it took some male guinea pigs, put them in cages with heating pads underneath to simulate climate change, and they kept them there for 70 days, the time it makes to make one cycle of sperm. Then mated them with female guinea pigs, and they analyzed the pups and the dads. And within that same generation, at least 18 different genes had different expression on them as they related to temperature regulation. And that's how they're able to tweak their own DNA. And so therefore, from a research program agenda for a real theory, you would have one. I'm looking for those engineering principles and those corresponding system elements, the sensors, logic, and response mechanisms in that guinea pig. This is what my research program would have me looking for, and I expect to find them, and I predict that we would find them. Another example is of these rove beetles and how they're able to track their environment. Everybody can see the dark brown beetle on the left, and they can see the ant on the right. But that isn't an ant. That is the rove beetle. And that beetle can morph into the shape of an army ant, which is its natural enemy. And nobody has actually seen this happen, but it happens on multiple occasions. And there on your screen, you can see an army ant and you can see a rove beetle, which is morphed to look and smell and behave just like that army ant. And we can track over time that these same beetles have been able to do this for at least 9 different types of army ants. Quite remarkable, quite incredible that they're able to morph into these different shapes. In Current Biology, which reported on this, proposed that the quote, as you can see there on the screen, these beetles were poised for mimecophilia, that is to look like an ant. And this near clade-wide pre-adaptive ground plan may underlie their repeated ant mimicry. And the American Academy of Sciences has Eureka Alert. They reported on this exact same thing, and they report that the discovery published in Current Biology provides evidence that evolution has the capacity to repeat itself in astonishingly predictable ways, completely contrary to evolutionary thinking. Another study in Current Biology reported on stick spiders in Hawaii. And there on the screen, you see a gold one that lives on leaves. But there's a white one which lives on lichen and a brown one which lives on tree trunks. And they found that, say, this gold one went to one of the Hawaiian islands, it can morph into the white and brown forms before the white and brown forms from the preceding island could even get to that island. And these researchers said, we usually don't expect evolution to be predictable. But Hawaiian stick spiders of the Rhamnus genus have repeatedly evolved the same distinctive forms known as ecomorphs on different islands. The spiders arrive on an island and boom, you get independent evolution to the same set of forms, this sort of rapid and repeated evolution. We can ask whether there might be an underlying mechanism that leads to the similar patterns of predictably repeated evolution in the course of adaptive radiation. That the Rhamnus spiders have some sort of pre-programmed switch in their DNA that can be quickly turned on to allow them to evolve rapidly into these successful forms. But how that process might work is still unclear. I suggest that they look for the sensors and the logic mechanisms and the output responses. Another paper reported on predictable evolution trumping randomness of mutations. And this was research on E. coli in separate colonies which were exposed to identical stresses. And they reported that although the mutations, the driver of evolution, occur at random, a study on the bacterium E. coli reveals that nature has can find the same solution to the same problem again and again. Identical mutations appeared independently in the test tubes. And they went on to say that the E. coli bacteria can acquire predictable mutations to adapt in changing environments. And then I have one final study, which was on fruit flies. And there are many, many, many of these studies reported in our paper. And this tracked 10 different populations of over 100,000 flies in each population over a season of just 4 months. And they continuously repeatedly sampled their genome for changes in allele frequency. And they found that all of the populations were essentially tracking with each other, as there were major changes in their allele frequency just over that summer period. And the research was done at Penn State, and they reported on that research of the rapid adaptation in the fruit flies that they normally expect to take a long time, but was actually happening very, very rapidly within just a few weeks. And to what extent are we talking about? They report that over the course of the experiment, which lasted 4 months, the researchers documented changes in 60% of the fly's genome. With this direct observation of swift and continuous adaptation in response to the environment, a phenomenon known as adaptive tracking, the biologists have established a new paradigm for how to think about this in new timescales. And one of the major researchers, Dr. Schmidt, mentioned that we see the populations are able to track differences in the environment. The populations were constantly evolving and shifting throughout the experiment. I would argue that these processes are taking place in a lot of different organisms, but they're harder to measure over the appropriate timescales. So we see that these organisms are able to attract their environments in highly regulated, rapid, repeatable, and with very, very targeted responses. All of this is fitting the engineered basis of continuous environmental tracking. It's far more consistent with that than random mutations being fractioned out in deadly struggles to survive. Now, after you develop a model, the next thing you need to do is develop new research in order to test those models in head-to-head tests. And continuous environmental tracking is really the first anti-Darwinian explanation, which is a thoroughly engineering-based description that's completely focused on the organism and focuses on non-random changes there. So what were we able to find? As we looked at these cave fish, which is what we're doing research on at ICR, we noticed that there is, and everybody can see this, there is a change in their pigmentation from the sided fish, which lives in streams, to the hypopigmented fish, which lives in caves. And these are the same species in a sense because they can mate with each other and have fertile offspring. So you ask the question, of course, what leads to the reduction in eyes and what leads to the reduction in their pigmentation? And evolutionists have an explanation for that, which is like all other explanations. And this paper by Borowski in 2018 mentions that they believe it's due to a loss of function mutation in the OCA2 gene. And then they believe that this is happening due to this, as I mentioned, loss of functions. And for reasons currently unknown, OCA2 seems to be evolution's favorite for pigment regression. Therefore, we can put a head-to-head test their predictions, where they would predict that this loss of pigmentation is due to random and broken genes. And it's overwhelmingly, according to them, a result of broken genetic pathways and due to a loss of function. Well, we would have completely different predictions. We would predict that these fish were engineered up front to live in these caves, and that the changes in pigmentation and many, many other changes that you would see are not due to broken pathways, but are traits that can be downregulated and upregulated in a very purposeful and regulated fashion. So this allows us to put a head-to-head test in a real research program and answer to the evolutionist challenge, which we had right at the beginning of this about 20 minutes ago. And so as we put this to a head-to-head test, we have to ask which one of these provides a better explanation. So here's a fish that we tested. Obviously, it's a blind cavefish, hypopigmented. This is a female in the natural environment which she would be at, actually in a cave. It's in our aquarium right now. But we subjected her to simulated sunlight with the same intensity, wavelength, frequency, durations as if she was now reintroduced back into a stream. And the evolutionary prediction is that her genes are broken, therefore she should stay in this hypopigmented state. Our prediction is that she would regain her pigmentation because it can be upregulated just as it was downregulated. And this is the fish after 32 days of sunlight exposure. Clearly, ability has not been broken. The ability remains there. And this is the male fish exposed at the same time. Both of these are still alive. They've just been stunned a little bit so we can take this picture. And this pigmentation can be regulated up and down as these organisms continuously track environmental changes. Therefore, we have two explanations. The evolutionary random adaptive process, which they say is not designed, which sees passive organisms shaped over time, and their changes are basically undirected, trial and error due to iterative processes, which Darwin said were numerous successive slight modifications, put to a head-to-head test against one which sees organisms as designed, with purposeful adaptive processes, not as passive, but as active problem solvers, and with mechanisms that are highly regulated, producing targeted solutions that are predictable and repeatable, just as I gave by all of these examples. And it is a system that can track the environments in many directions which it can go within its design parameters. So I think we've pretty much taken apart the theory, and we've offered a viable additional theory as an explanation for that. And so, as I mentioned, you can get more information from the paper that I had at the beginning of this talk, or you can turn to our own webpage, as Dr. Thomas already mentioned, at icr.org, and tune into any of these podcasts and get more additional information. So I know we covered a lot of information in a short amount of time, but thank you. Thank you very much for enabling me to go through this and hopefully explain it for all of our listeners.
Dr. G, that was fantastic. That is real science right there. Your podcasts that are put out on the ICR YouTube channel are excellent. I really enjoy those. So great work to your team. And what I kept thinking was, when you start with a biblical worldview, you can make these types of accurate predictions on what to expect in the biological world. And so excellent job, Dr. G. That completes our second presentation for tonight's Science Confirms the Bible seminar. I will bring in our next speaker, Dr. Dan Biddle from Genesis Apologetics. Certainly no stranger to standing for truth. Not your first time here, Dr. Dan. So it's great to have you back. Dan, I'm excited for your presentation or talk titled Top Evidences for Noah's Worldwide Flood. We are going to have Sam give you a formal introduction. As you get that ready, Sam, I'll remind the audience, as we are doing this live, we're well over 200 people in the live chat enjoying this. We will be having a roughly 20-minute panel discussion or audience Q&A once we're done with our 4 presentations. So please, if you do have a question or even an objection, just make sure to send those in a professional and respectful manner, and we will have some fun engaging and interacting with your comments. Sam, if you'd like to give our guest here, our third speaker for the night, a formal introduction.
Absolutely. Dr. Dan Biddle is the president of Genesis Apologetics Incorporation, a 501c3 organization dedicated to training youth pastors, parents, and students about Genesis, creation, and the flood. Daniel has authored and edited 12-plus books and several articles on these topics, produced 3 films on Genesis and apologetics, and is working on a 4th, The Tower of Babel, coming to theaters in 2026, produced over 100 training videos on the Genesis Apologetics YouTube channel with 223,000 subscribers, more than 23 million views, and content translated into 13 languages. Daniel has given hundreds of presentations to thousands of people on these topics. Daniel's professional background includes an undergraduate and graduate work in theology, apologetics, training as a behavioral scientist, a PhD in industrial organizational psychology, and 20 years experience in expert witness consulting and testimony in state and federal cases involving scientific research methods, statistics, and psychometrics. Daniel and his wife, Jenny, live in Folsom, California, and enjoy their 4 adult children. While Daniel has been a Christian since the age of 11, his position on the specific chronology of Genesis has remained undeclared until 2011, when the evidence surrounding the fossil record, dinosaurs in particular, flood geology, and his biblical exegete led him to the historical position on the Genesis account.
Sam, appreciate that. Dr. Dan, how are you tonight?
I'm doing great, you guys. Thank you so much. I am excited to be here. This is too much fun.
Amen. I completely agree. Let's get right into it, brother. I'm excited for your talk.
I will pull up your PowerPoint here and let me know when you can see that full screen.
We are good to go. The floor is yours, Dan.
Okay, terrific. Well, you guys, thank you again for having me and Dr. Thomas and Guliuzza. Those were fantastic presentations. We love what you guys do at ICR, and we're glad to share the stage with you guys. So just to start out, this is going to be a very quick tour, about 20 minutes worth of evidences. If you want the deep take on Noah's flood, we have a 2-hour film that was in about 1,000 theaters last year. It's now free on YouTube. You can just go to noahsflood.com or pull it up on YouTube by just searching for The Ark in the Darkness. It's currently free. And here's why this is important. This is a plaque that you can find at the Creation Museum at Answers in Genesis. They have a great ministry and a great full-size ark in the middle of America that you can go see. It's incredible. So if Satan were to say anything about the flood, he would say this: if I can convince you that the flood was not real, then I can convince you that heaven and hell are not real. And today it's not some, it's not half, but most of Christians graduating, at least in California, from high school are being converted into theistic evolutionists, believing that God used a long, slow, clumsy, murderous process to bring about life on earth. So the stakes are high and the significance of the flood is very real. So let's take a look at the first evidence, and that's that flood evidences or flood legends are everywhere. Here's one table. There are many different ones that we can share, but when you take a look at all of the different cultures around the world, there's over 300 of them that have flood myths and legends, and many of them you can put out on a grid work like this that have similarity, like there was a destruction by water, the humans were saved, the animals were saved, there was one guy that God or the gods appointed to do it. This is just one chart. There are many like this, but there's at least 300 worldwide flood stories from around the world, and they have remarkable and striking similarities that overlap the actual Genesis account. I mean, it's remarkable. You can even look at the Chinese ancient writing, and to say the word or write the word boat, you would say large ship with 8 people. And of course, that's very reminiscent of the Genesis account where you have 8 people on a large ship. Um, that's one book by Bill Cooper that's come up with all kinds of evidences from a worldwide perspective about all the different flood legends. Another one of my favorite, is by Nick here. This is a book called Echoes of Ararat, where he's compiling all kinds of different flood legends and myths and stories around North America and South America that have echoes from both the flood account and the Babel dispersion. And you guys, it's a great read. It's really incredible how much evidence there is from a historical basis about a worldwide flood. Every single dot that you see on this animation here is a culture where a flood myth, an ancient flood myth or legend, has been documented. And look what happens when you spin the globe and get to North America. You guys, the Native American people carried with them a very clear account of the flood after the Babel dispersion, just thousands of years after. They're in North America, and you go interview these tribes, and they have remarkable histories about the flood, and some of them have an uncanny parallel to the Genesis account. When we look further or more closely at the ancient Near East, we have all kinds of Babylonian flood accounts. We have the Eridu tablet, the Gilgamesh epic, the Simon's Ark tablet, the Atrahasis epic. The Middle East, which is of course where the Ark landed and where the Babel dispersion happened, has all kinds of incredible, very ancient accounts about the flood. Here's what they look like. A number of them are etched out on cuneiform or clay tablets, but they have incredible overlaps to the flood. And one of the things you'll notice when you look at these epics and these ancient Near Eastern mythological accounts is that the further you go back in time, the more mythical accounts will get. It doesn't work the other way. At the very, very root of some of these, you're going to have the truthful account, which is a Genesis account. But these stories don't tend to get more truthful over time. They tend to get more mythical. So when you reverse engineer and go back to these ancient flood myths, they have an incredible origin or root in the Genesis account. And they all seem to draw on these traditional elements, things like there's a God who's going to bring punishment to the world. There's one chosen guy to save everybody with his family and the animals. There a God or gods are going to give a dimensions to a ship that's going to save everything. They're going to survive the flood. Many of these accounts actually have that he's going to send a bird out and to find dry land and have sacrifice to God afterwards. So isn't it interesting that these ancient Near Eastern myths and a lot of the stories carried over by the Native Americans all still have these same common themes and elements? When you look at ancient writing that include, of course, a lot of these myths, it's interesting that if you interview experts on cuneiform writing, like the curator of cuneiform from the British Library, they'll say things like, when you really press them and say, well, how old are these writings really? How old are they where you can confirm for sure how old they are? They'll give you a bracket saying about 750 BC to about 1500 BC, because the people recording these events can correlate them to observe things that we know happened on Earth during those times. When you start going back further, you have to start borrowing faith. And of course, isn't it interesting that humans have been smart for a long time, but all human writing vanishes about the time the Bible puts the flood? That's certainly no coincidence. You can look at Hebrew writing, Chinese, Egyptian, ancient Greece. It all vanishes, so we have to use faith. We have to evoke faith for what we're going to fill up in the time before the flood. But I find it fascinating that all human writing in history vanishes about the same time the Bible puts Noah's flood. Let's look at the next evidence here is the feasibility of the ark. When you compare the ark and the dimensions that God gave to Noah for building it to the Epic of Gilgamesh, we immediately see all kinds of differences. The biblical ark had 50 to 75 years to build it. It's over 500 feet long. The duration of the flood was a year, and it's definitely seaworthy. You can contrast that with the Epic of Gilgamesh ark, where the flood was only a week. It only took a week to build the cube-shaped ark that had 6 or 7 stories to it. It's 200 feet on each side. The first rotation at sea is going to kill everything, so it's definitely a mythical account. Then you have the Simmons Ark tablet, same thing. You've got a circular ark, which is also not very seaworthy. But when you look at the ark itself and you run hydrodynamic testing to it, it definitely was seaworthy. We've hired maritime experts and engineers to model the ark and show that you can crank up what's called a Beaufort storm scale to level 7, 8, 9, 10. You cannot get the same to capsize. It would hold up to all types of drama out in the ocean. But when you start rotating the Epic of Gilgamesh, it would definitely fall right apart. It's even been modeled here in the early 90s at the Korean Research Naval Center. They tried 12 different models, and it was ideal. The dimensions of the ark were ideal with respect to stability, comfort, and strength. So here we have the Epic of Gilgamesh ark, and when you crank up the storm scale to 8, 9, or 10, it's going to start rotating. Same with the circular ark, but the Noah's ark, that using the dimensions that God gave, is going to hold up. In fact, it can even go back and forth to up to 45 degrees, and it would still right itself. So we have an ancient biblical account with an ark dimension that could actually work. Number 3, let's look at unimaginable numbers of dead dinosaurs mixed with marine life. Here's the most obvious thing about the flood. If you look in the middle of America and you plot out the Jurassic and Cretaceous fossils from Fossil Works here, you have an amazing, huge dinosaur kill zone. It spans 1,800 miles up and down, about 1,000 miles wide, 1 million square miles of a dinosaur kill zone. And these creatures are buried in a matrix, mostly of 3 products, mud, sand, and ash. And we'll talk about that more in a little bit. But here is some really huge, obvious evidence for you. How in the world do you bury dinosaurs and mix them with marine life over a quarter of the landscape in America? So let's take a look at one of these areas called the Morrison Formation. It's a swath of 600,000 miles encompassing about 13 states in the middle of America. If you take a cross-sectional look at where we just zoomed into, which is Morrison, Colorado, you'll find that that 600,000 mile surface area called the Morrison Formation is actually a 300-foot thick mud pancake that incorporates over 50 dinosaur species that all bought it at the same time. Most large forms of dinosaurs in the Jurassic are all found buried there. How did they all die at the same time and get jumbled up and mixed up with marine life? Isn't that quite fitting to the flood? All kinds of fossil occurrences, different genre, many different species, and that's just what we found. And they believe we only know about the top 20% or so of the Morrison Formation. So 600,000 square miles are filled with these creatures, and it took a flood to do it. Number 4, Dr. Thomas covered this. I'm going to share just a few of my perspectives because this was one of the most convincing things that shifted me to becoming young Earth creationists. And as Brian mentioned, we're now up to 127 peer-reviewed secular science journals that has established not 1, not 2, not half a dozen, but 16 different types of bioorganic materials that are found in dinosaur bones that should not be there. Mark Armitage took a triceratops horn, demineralized it, and it still has soft, stretchable pliable material in it. You should not be able to do this with a Triceratops bone that's millions upon millions of years old, but yet you can. Very, very interesting. So over 120 peer-reviewed journal articles, over at least 16 different types of bioorganics, but the last two that they found are, get this, cartilage and nerve cells. How on earth can you fossilize and preserve dinosaur cartilage and actual nerve cells for millions of years? You can't. You guys, the evidence is on our side. It's case closed. These materials are young and they are fresh. The biggest one that I find most convincing, as Dr. Thomas went over tonight, is collagen, because I've read the literature. Dr. Thomas is a scholar in the literature, and I found at least 5 different scientific studies that have tried to estimate the maximum lifespan of collagen. And lo and behold, as Dr. Thomas said, the most they'll give it is about 900,000 years. So you draw the cap at about 1,000,000 years. So how on earth is dinosaur collagen lasting in dinosaurs if they're really 65 million years old? When you run the math, it's about 100 times longer than current science journals say it can last, but it's still there. So it's really easy to shift this evidence over to our perspective and say that these bones are just thousands of years old, not millions of years old. Next, let's take a look at the rapid catastrophic burial of these dinosaurs and how they're mixed with marine life. Here's an interesting article from a secular paleontologist, Dr. John Horner. Look at the subtitle of his book here. It's called Digging Dinosaurs, but the subtitle is quite interesting. He says, "The Search That Unraveled the Mystery of Baby Dinosaurs." And here's a quote from his book. He said they had discovered one huge bed of Maiasaur dinosaurs, stretching 1¼ miles going east to west, which would, of course, fit the flood, because we believe that the sheet layers that are deposited in America are from tsunamis that are coming up from the sides of the coast. And he says, look, there were up to 30 million fossil fragments in that 1 mile just over an area. They had discovered the tomb of 10,000 dinosaurs, 10,000 Maiasaura. But here's the catch. There's 10,000 of them, representing 30 million fossils spread out over a mile directionally, and they couldn't find a single young. Every single one of the 10,000 dinosaurs that they found were between 9 and 23 feet. So what on earth could cause a panic of a herd of dinosaurs, 10,000 of them, to scamper and run and ditch the babies, the youth, the adolescent, the juveniles? They're not around. These things booked it, and they were all found buried catastrophically in a flood. I find that very interesting about how quickly this really happened. Dr. Werner has also done a book I'm going to show here in a minute, but here's another one about bone beds. Dr. Werner's book has established that of 100,000 dinosaur fossils that have been found, only about 3% of them are articulated or found complete. 97% of them are found scrambled and disarticulated because something catastrophic hit them. In fact, you can find in the Lance Formation locations where there's huge dinosaur burials that are in sorted beds, and the experts there would say these dinosaurs were hit and killed by a tsunami miles away and then moved and reinterred over here where they were buried. So something happened quick and quite catastrophically. Dr. Werner's book has got a great way to look at that. And frequently when you go to these museums, you'll see these dinosaurs, theropods particularly, in the dinosaur death pose with their necks arched back as they're suffocating, gasping for air. And that's because they were killed by the mud that they're entombed in. They didn't just die and lay down and get covered over by thousands of years worth of mud or atmospheric dirt. No, they're actually buried in the product that was responsible for their death. More on that as we go. And it comes down to these 3 products: mud, sand, and ash. How in the world do we find these dinosaurs buried in those 3 products? But that's what we find. When we dig in many different places around the United States, we find that these dinosaurs are buried in a matrix of those 3 things. It's very indicative. It's a telltale sign of how they died, and it happens to fall right on top of a biblical explanation. So the interesting insight about all these different accounts that want to talk about the flood and ancient histories and everything, I'm going to say that the Bible's the only ancient text that describes the processes of the flood. It's got timelines, mechanics, it's got how high the water came, when it came, it's got the 40-day events, 150-day events. It's got a whole narrative there of the flood process, and it actually gets it right, which is incredible. You can't say that about any other ancient text. It's incredible. So here's where the key verse comes in Genesis 7:11. It's the 600th year of Noah's life, and the second month, the 17th day of the month, the same day were all the fountains of the great deep broken up, and the windows of heaven were open. So something happened. It started on the ocean floor, and it began, and then later the upwards, the water came all the way upwards, was about 22 feet over the highest hill in its zenith at day 150. So we have the flood mechanics and the processes all laid out in Scripture. And I also find this verse very interesting. One could say, when you look at this verse carefully, it's talking about water going forth and returning, like sheets coming up, sheets coming back off. And it could be an indication of what we know happened during the flood, tsunamis, multiple tsunamis happening in cycles. Dr. John Baumgartner came out with this model called catastrophic plate tectonic, he and 5 other PhD scientists. He developed a software system that was based on it, showing how Pangea or a Pangea-like formation was rapidly split apart as the fountains of the Great Deep broke open. This has been studied. Layers of research have been done on this. The model checks out. And that's exactly what would produce these 3 products that we find the dinosaurs entombed in. When the fountains of the Great Deep broke open, it's going to produce mud and sand from the subduction, and the subduction are going to create a whole bunch of slip faults that are going to happen as the newly produced seafloor dives underneath the land continents, breaks loose, and it's going to cause a tsunami to come up on land. And it's that same rapid subduction of the new seafloor that's going to cause volcanism, that's going to be burying the dinosaurs as these tsunami cycles are happening in waves and repeated cycles. So here's what it might have looked like when the fountains of the Great Deep broke open. We have underwater rifting here. We have the lava coming up. It's going to create these huge supercritically heated steam jets that are going to come up, bring up waves of tsunamis. And we can see this on Earth today. It's a 40,000-mile rift system, produces the new seafloor. It comes and hits the land mass. It's going to bind, develop tension, and then launch water onto the Earth in cycles over and over again. Here's another image of that new seafloor being created. It's going to come, it's going to be like a conveyor belt. It's going to spread the new seafloor on both sides. It's going to hit the land and then build up tension and create a tsunami. So here's that new seafloor, hits the landmass. It's going to bind just like what happened in Japan, where there was a 60-foot seafloor slip that resulted in tsunamis. When that tsunami happens, it's going to come up and wash these dinosaurs and bury them in layers. And that's exactly how we see them buried today, in layers full of mud, which is exactly what a tsunami would do. So where did the ash come from? Well, same thing. We have the subduction happening. We have volcanism along the continental margins, and it's raining down on these dinosaurs. And we can look in several spots where what's called the Independence Dyke Swarm, which is a 375-mile linear rift system that produced 4,000 cubic miles of ash and blanketed half of America. Huge amounts of ash came and buried over, and enough to bury parts of entire states. You guys, it's just incredible to look at this, and it's deeper than you can imagine, over 100 meters deep in some region. But that volcanic system is responsible for burying the dinosaurs with mud and sand concurrently, and that's why we find them buried in those 3 products. So just the Brushy Basin member produced ashes 110 meters thick in eastern Utah and 35 other locations, and it's filled with dinosaurs. So by comparison, you can see how much ash was produced during the flood. When the Independence Dyke Swarm broke open, the smallest square over all the way to the right is how much ash was released when Mount St. Helens blew its top. By comparison, it's just almost unimaginable how much ash came out of that system during the flood. Here we see layer after layer of the ash layer that you can see here with mud and sand, just it's like pancaked right on top of each other, and we find dinosaurs buried in all of these layers. And so these dinosaurs were killed and buried by the processes and products that bury them. They didn't just, go out to eat, have too much to eat someday, get a disease or whatever, and lay down and get covered by atmospheric dust. So they were killed by the products that they're buried in. The next one is we'll look at how these massive oceanic plates were dragged under the continents. We know this happened. Here's an underground radar that was done of the Farallon plate. We can still see it subducted. This was responsible for a lot of the volcanism and a lot of the tsunamis that came up on the North Americas. Here's the conjugate Shatsky Rise, which is an oceanic plateau that was pulled from the southwest there and drug underneath America, causing a lot of subduction related activity and a lot of mountain building. Some of it happened during the flood, but a lot of it happened afterwards. So the seafloor that's being pulled underneath is what's going to be causing a lot of these tsunamis. Brian touched a little bit about on this, but when you look at the fossil record, isn't it interesting that the first three Sloss megasequences, the Sauk, Tippecanoe, and Kaskaskia, are mostly marine life? It's not until you get much higher that we start seeing land. This is exactly, or land creatures, this is exactly what we would expect and predict with the flood. You would have the marine life in the shallow seas buried first, then the land creatures are going to be the next ones to have it. And that's exactly what we see these Sloss mega sequences happening. We don't get land creatures until we get to the Absaroka and the Zuni, just as Brian talked about in his presentation. Here's a fossil distribution, a frequency chart showing all the marine life in those first three mega sequences. Then when you go up, that's when all of a sudden we start getting all these land creatures deposited. It's perfectly fitting of Noah's flood. And that's because, again, we're going to have all this new seafloor going. It's going to bury these sequences first, and then it's going to come up on land, and that's when it's going to be responsible for burying the creatures. It wasn't until the Zuni formation that we start seeing at the very, very top all these land creatures getting buried. It was the zenith of the flood at day 150. That was the dinosaurs' last stand. Just a few more things here, and then we'll close out. Evidence 9 is correlated fossils and oil. Isn't it interesting when you take these continents, and we know that they're perfectly matching when you fit them back together? Because remember, the Bible says that on the same day, all the fountains of the great deep broke open, and that was the active force that pushed these continents apart. Each one of these little dots is a fossil bone bed, and when you put the continents back together, we can see entire ecosystems where you can see a brother buried on one side of the continent and its sister on the other side, cousins buried on either side of these things, now separated by thousands of miles. But when you match the continents back together, the same flora and fauna is buried in the different mud systems that push them apart, but they perfectly correlate when you put them together. You can run the fossil counts and all the data is there. And isn't it also interesting that the oil on each sides of these continents is actually correlated? The chemistry of the oil indicates that the same type of ecosystem, the same biosphere, was being buried during the flood when these continents were pushed apart. Very fascinating evidence here. And the last one that we'll look at is the receding stage of the flood, and that's when God said he shut off the fountain. What that's going to do, the cooling effect is going to pull the water back off of the continents, first in sheet flow and then in channelized cuts. And that's exactly what we see in places like now the Gulf of America, where we have what's called the Whopper sand. Dr. Tim Clary with ICR has done tons of study on this. We have all kinds of runoff from the Tejas major Sloss mega sequence. And we have the Monterey Bay underwater sea canyon here that's about the same size as the Grand Canyon, but it's all underwater, and it happened from catastrophic water that was sheeting and then flowing in channelized cuts off of the continents. So I think we have evidences on a number of different perspectives, and I think we just gave a raced tour through them tonight, but I personally find them quite quite convincing. So we'll close out there and do the Q&A at the end, I guess, huh?
Dan, we definitely titled this seminar correctly, Young Earth Creation, Incredible Evidence. That was a great presentation.
Thanks.
3 information-packed presentations so far. We are now moving into our 4th and final presentation for tonight's seminar. I will bring in our 4th speaker, Dr. Dan Biddle. Appreciate that presentation. We'll bring in Dr. Carl Werner. It's great to have you back as well here on Standing for Truth. Certainly not your first time here as well. We did a 3-part series together on living fossils and how human evolution has been overturned. Fantastic series. So Carl, it's great to have you back. And I'm excited for your presentation and what we'll do before we get into your talk on worldwide fossil evidence that proves creation and the flood. We're going to hand it over to Sam. Sam, if you could, we'll give Dr. Werner here a formal introduction.
Absolutely. Thank you. So Dr. Carl Werner is an award-winning filmmaker and evolution theorist. He graduated from the University of Missouri with the distinction in biology, summa cum laude. He traveled an impressive 298,000 miles to 45 dig sites in 16 countries around the globe, photographing thousands of original fossils and the actual fossil layers where they were found. He and his wife took more than 250,000 photographs, and he also interviewed 100 top evolution scientists at 120 top universities and museums. He is the author of a 5-volume Evolution, the Grand Experiment book series, and executive producer of the 8-episode Evolution, the Grand Experiment video series. He received his doctoral degree in medicine at the age of 23.
Sam, thank you very much. Dr. Werner, how are you tonight?
Doing good. Thanks for having me, guys.
My pleasure. Let's get right into it. I'm looking forward to your presentation as well, Carl. So allow me to get your slides up here, and the floor is yours. Go ahead.
And I just want to say to the audience, you have to pay attention because I'm going to be asking questions for you to answer in the chat. So get ready with your keyboard, and you're going to have to participate by thinking. So if you stand at the edge of the Grand Canyon, it is an awe-inspiring experience to look down at the very bottom, the Colorado River, and see 1 mile of sediments. And you think, how in the world did all those sediments get here? Well, I'm going to pull back the curtain and show you the 2 theories. One theory is that all the layers of the Grand Canyon were deposited over 1 year during a tsunami-based submersive worldwide flood. The other theory is that the rock layers of the Grand Canyon were deposited over 1 billion years, not 1 year, but 1 billion years, from periodic inland seas, deserts, and rivers. And the theorist in evolution would say that the Grand Canyon is just part of a bigger structure called the geologic column, which is on the right here. Now, there's no place on Earth that you can actually see the geologic column, and I'm not sure that it really exists. And we could discuss that another day, another time. But I just want to show you the layers of the Grand Canyon. There's only 6 layers, and they only comprise these 6 layers of the geologic column. And in fact, 2 of the layers are missing right in the middle of them, which is odd. But to create this whole geologic column, you have to go to other places in the world, like the White Cliffs of Dover. Those are Cretaceous layers. That's how you get that layer there. Or the Negev Desert. That's how you get the Triassic. Or the Fossil Butte, Wyoming. You can get the Eocene fossils. But you have to assemble this by going to different locations. And I always say if you can't see something, you have to question whether it's real or not. So this is an either or situation. Either the Grand Canyon sediments were laid down over 1 billion years or 1 year. Wow, that's pretty different. You should be able to easily disprove that. So my first question for the audience, and this is for the evolutionists in the audience only, the creationists I don't want you to answer yet. how many layers in the geological column out of the 18 layers, the Holocene be 1, Pleistocene, there's 18 of these. How many layers have saltwater creatures? Now, I want you to put your answer in the chat as identify yourself as evolution, and you could say 5 layers, 15 layers, 18 layers, whatever you want to say. But, like, for example, the Cambrian, we all know that there's trilobites in it. That's a saltwater creature. And the Mississippian has crinoids in it, and that's a saltwater creature. How many of these layers have saltwater creatures? Now, for the creationists in the audience, I have a different question, is how many of the named rock layers, these 18 named rock layers, have saltwater creatures? It's basically the same question. For your answer, I want you to put in chat creation. I think there's 10 or 15 or whatever. But I want to caution those who believe in creation what is actually taught. Noah's worldwide flood was an ocean-based, or you could say a saltwater or a marine-based flood. It wasn't freshwater flood. These were the oceans of the earth crossed over the continents. So you would think that in Noah's flood, there would be saltwater creatures like sharks and starfish and nautilus being mixed up with the land animals like T-Rex or snakes. So my question to you creationists is, how many layers should have saltwater creatures? There's 18 layers of rock, theoretically. How many should have saltwater creatures? So Donnie, I'm going to ask you to read the answers and tell us what the answers are quickly.
Carl, the answers that all came in are saying all, mostly creationists, but I think it looks like with the evolutionist side, it would say all as well.
Okay, so, and that is correct. There is saltwater creatures in every named rock layer. Now, that's news to some people in the evolutionary community, but the saltwater creatures in every layer is predicted by the flood model. And this would be like a trade secret in paleontology. Now, I'm going to go through them. I'm going to prove that to you. This is a sea pen. I filmed this at an aquarium. It's a saltwater creature. It's living. And this is a fossil sea pen. And where was it found? It was found in the Ediacaran layer, the lowest layer. The next layer up is this fossil sponge. Sponges are alive today in the oceans. It's a saltwater creature. Where was it found? In the Cambrian. This is a long stem crinoid. It washed up from the ocean on New Zealand coast. Debbie picked it up. And this is a fossil crinoid. It's a saltwater creature and is found in the Ordovician. This is the Great Barrier Reef. And here's a coral. Corals are in the ocean. Here's a fossil coral. And this fossil coral was in the Silurian. So as we walk up, I want to show you that there is, in fact, saltwater creatures, starfish, corals, sharks, shellfish, saltwater snails, saltwater rays, saltwater sea biscuits, saltwater sharks, saltwater sand dollars, shark teeth, crabs, sand dollars, coral, and saltwater shells. So every layer has saltwater creatures. But here's the problem. All those fossils I just showed you, here's where they found them. And like, for example, this coral was found in Illinois, and the elevation there is 800 feet. That means that the water had to rise to 800 feet higher than it is today for this layer. And this one here in the Burgess Shale, the salt water had to rise 3,000 feet. Right here in Arizona, these saltwater creatures are thousands of feet above the level of the sea today. This would mean that the seas would have to cross and come onto the continents 18 different times for these 18 floods. See, these are all separated by millions of years, and we know that there's land animals in many of these layers. So you have to explain how you could have this many floods, and this isn't a quarter of an inch rise of the ocean levels from global warming. This is 1,000 feet, 2,000 feet, 3,000 feet in every layer. And so you're, as an evolutionist, you're confronted. there would have to be 18 worldwide floods because there's saltwater creatures in all 18 layers. But the creationists would say, no, there's just one flood. And which is more believable? It's hard enough to believe one worldwide flood. You know, where did water come from? Where did it go? Et cetera, et cetera. But 18 times or some number, 15 times, whatever number you want to come up with, that's more ridiculous than Noah's flood story. It's so unbelievable. My next question for the audience is, how many layers in the geological column out of these 18 layers over here have modern animals in it? The top layer we believe has, evolutionists believe has like modern mammals in it, but the bottom layer would have these strange and unusual creatures of the Cambrian, the Precambrian called the Ediacaran. How many of these layers have modern animals been found in the layers? So if you're an evolutionist, I want you to answer it. Evolutionist, I believe only the top 3 layers or 5 layers or whatever. And if you're a creationist, the same question, how many layers have modern animals in it? And this is very important. And I want to guide the creationist to what the flood model would predict. See, according to the biblical account of creation, God made Adam and Eve in the Garden of Eden. And at the time, there was a snake that tempted them, the apple tree. And of course, there'd be a flamingo and a duck and some pretty flowers. But there was more to that scene, because the scripture says that God created all the animals and plants that have ever lived on earth then. So that means that Adam and Eve would have been living with all the extinct animals that we never see anymore, like dinosaurs or flying reptiles. And so all the animals living and extinct lived back then, and they were all present. So if that's true, if you had back at that time before Noah's flood, living animals that we know of, familiar animals, with extinct animals that we're not familiar with, and they were all mixed up in a global flood, and they were all living together, how many of the layers should have modern animals in it? Starting with the top layer all the way down to the bottom, how many? So, Donnie, what are the answers for that for evolutionists and creationists?
The great majority have said all, with at least one saying, I bet the top 15 have modern animals.
Okay, so the answer is that modern animals are found in every named rock layer.
Wow.
And that's predicted by the flood model. And it's another trade secret of paleontology, because these charts don't show you that these are modern animals in all these bottom layers. And I need to clarify really quickly, modern versus modern species versus modern appearing. This is a bunch of dog skulls. They're modern because they were just recently died. And they are the species Canis familiaris. Genus is Canis familiaris as a species. Binomial name is Canis familiaris. And look how different a Chihuahua skull is from a bulldog skull from an Irish wolfhound. The Chihuahua skull is smooth and small, and his teeth go straight down. The bulldog has these big, wide bumps on top of the skull, and the teeth go out to the side, whereas an Irish wolfhound is a very long skull, and the teeth go down. If you had never met a dog before, you would think these were different genuses and species, but in fact, all these animals can reproduce with one another, so we know they're the same species. Well, same with this living nautilus. It is living today, okay? And it is called Nautilus pompilius, genus Nautilus, species Pompilius. And if you cut it in half, this is what the inside looks like. Now, they found a nautilus back here in the Jurassic layer and cut it in half, and this is what it looks like. But they changed the name of the fossil. If you put the fossil next to the modern, it looks identical, even down to the siphuncle, which is a tube which equalizes the pressure. You can see the siphuncle in the fossil, and you can see where the siphuncle lays in the modern. It's fleshy, so it doesn't stay. But look what they did to the species and genus names. Even though it's the same animal, they changed the genus name and they changed the species name. So you cannot trust evolution scientists on any of these names, because they rename everything. And I'll show you some examples of this. Every fossil I'm going to show you has a renamed fossil name, and they put them in a different genus. But these two look more similar than two dog skulls, and the two dog skulls varied greatly. And these are of the same, they look like they're the same species, but of course they call them a different species. This is a worm, a bristle worm that was found in the Cambrian. Now, that doesn't mean much to you, but bristle worms are very important. They're very emotional for Debbie and I. This is Debbie when we were filming in the Caribbean, and she just got back up onto the sailboat, and she did not look happy. And I said, what happened? She said, well, a bristle worm got onto my face right above my snorkel, and it stung me, and my lip is swollen. It all burns. This is all burning. And so this is a bristle worm that stung her, not the particular one, but the species that stung her. Okay. So this bristle worm down here was found in the Cambrian, and here's a picture of it. And this is Smithsonian. Here's a Smithsonian drawing of what that fossil bristle worm looks like. And look at this. They look like they're a perfect match. Of course, they changed the species and genus name, but you can say the bristle worms were in that layer. The starfish look the same in the Ordovician. If you look at this one, the underneath surface, these two like side by side, and you see the side by side holes here. This brittle star is from the Devonian. Now, color doesn't get preserved in fossils generally. So here's a brittle star in Hawaii. But if you just take the color out, you could see these look closer than two dog skulls. Here is a sea lily that was found in the Mississippian layer. Now, here's Debbie. She picked up this sea lily, crinoid, long stem crinoid, on the coast of New Zealand. It just washed up from the Pacific Ocean, and we started filming it. And look how close this thing is to this. It even has the long stems. I always thought as a kid the crinoids were extinct, but this looks like it's the same species. You don't know that for sure, but it looks as close as two dogs, certainly. And so you would say this is a modern appearing fossil, could definitely be the same species. Here's a starfish. There's a modern starfish. Here's from the Carnegie. I'm going to show you, there's three fossils they had on display for this layer. The modern looking dragonfly, the modern looking cockroach, the modern looking scorpion, and this modern looking lungfish, all found in this Permian layer. And if you look at the Permian fossil drawing at the Carnegie and a modern living lungfish, they look like exact copies. And I'm going to go through these quickly because I'm going to run out of time here. But there's modern animals in every layer, including frogs. And this is my favorite, this coral. So in summary, there's modern types of animals, saltwater animals, and extinct animals are found in every named rock layer, all predicted by the flood model. And my third question to you is, how many river channels can be seen in the rock layers of the geological column? And I want hundreds, thousands of millions. Evolutionists, put your name in evolution, and you think, how many times can you see a river channel in these layers? And for the creationists, how many river channels can you see in these rock layers? But I want to caution the creationists, there would be no rivers in Noah's flood because this is a big single water event. There's no rivers below this boat here. There just were no rivers when all the rock layers were deposited. So we would expect no rivers. So what were the answers, Donnie, for the evolutionists? How many river channels can they see in the rock layers?
That one may have been more of a challenging question, Carl, because I'm not yet seeing
no worries.
Any answers.
No worries. So I'm going to show you something. There are no river channels are found in every named rock layer. There's none. And how do I know this? This is going to take some thinking on your part. You're going to really have to put on your thinking cap with the gyrations of your brain showing. This is going to be hard to explain to you, but you could rewatch this show later. I'm going to do one. Here's at Petrified Forest, Arizona. Hundreds of thousands of trees were buried by a river in flood. That must have been a big river, okay? Yet, all the layers of petrified forest are flat. There are no river channels. You would expect river channels if this was over millions of years. See, all the layers are flat. Now, if evolution would predict that before the time of X, if you removed all these layers in time and go back to just this layer, that the surface of the land would have been flat at that time. The white layer, this would have been a flat surface below the X. If that was true, there should have been river channels created when none of this was here above air, and it was just streams going through this layer. And then as the next layer gets deposited, and the next layer here is this brownish sediment, it should fill brown sediment into these channels that were formed in the previous time. And so you should see these kind of river channels all throughout this area. But this is a Photoshop. There are no river channels at petrified forests. That's what the reality of what this looks like. There are no river channels. And if you step back, wow, no river channels. It step back even farther, no river channels. You have to inspect them close when they're irregular like this. Same thing happened at Dinosaur Provincial Park, 10,000 dinosaurs buried in a river. You would think there'd be river channels all over. There's no river channels at petrified forest, just straight flat layers. Zion National Park, no river channels. Joggins Cliffs, no river channels. Road cuts you see all around the world. And this is worldwide. There's no river channels. So this confirms that the sedimentary layers were deposited underwater by a worldwide flood. And I think, Donnie, I've reached 19 minutes, and I want to be respectful. I could pick up a couple more topics here, but I think I'm going to be respectful of time. And oh, see, there's my alarm. So I'm going to say we should call it a day and move on to the question and answer. If I can figure out how to turn off the sound here.
No worries. Time flies by when you're having fun, upholding the truth of biblical creation. And so let me bring in my co-host here, my award-winning co-host, and Dr. Carl Werner. Thank you very much for that presentation. Time flew by. I could have easily listened to another 2 hours each on every single one of those presentations. And so we do have enough questions to keep us busy until same time tomorrow. Therefore, let's bring in all of our panelists, Dr. Brian Thomas, Dr. Randy Guliuzza, Dr. Dan Biddle, and okay. Well, that concludes our 4 sessions, Young Earth Creation, Incredible Evidence. I said when we first started promoting this, that this seminar will show exactly why it is a great time to be a biblical creationist. And you brothers did not let us down. Excellent work. Sam, what were your thoughts on those presentations, brother?
Yeah, that was fantastic. I'm thank you for letting me be here, be a part of this. So thank you, gentlemen, for your time.
Welcome.
And work. Time and work. Okay, well, let's get into some questions. What I think we should do for our roughly 20 minutes of questions here. First, I do want to let everybody in the audience know that in many ways, this was a sneak preview into all of these many lines of evidence for young Earth creation and the global flood. So if you do check the description box of this video, which I do encourage you to do, I have all the relevant links to ICR, to Dr. Carl Werner and Dr. Dan Biddle and Genesis Apologetics, where you can find more from them. Their YouTube channels, their websites, really just for endless material that you can benefit from. Okay, I've got a couple questions, it looks like, for everybody. And so why don't we start with our first presentation, which was you, Dr. Brian Thomas, on dinosaur soft tissue. Some truly fascinating information there. And I think this is a good question. This one came in from Apologetics 101 for Brian. For Brian from Brian. Brian, thank you for the question. What is your take on some of the responses to the blood cells and tissue found inside dinosaur bones? In other words, Dr. Thomas, obviously there's still people out there who are not young Earth creation. They still believe in the billions of years paradigm, but they are aware of this evidence. And so in terms of how they've responded or dealt with that evidence, what have been your feelings or thoughts on that?
Well, I'm going to assume that this question really has to do with some of the stories that have come out, or I may say generously call them explanations as to how you can get original tissue or original proteins to last much longer than they can last based on biochemistry. And so that's so that's, I think that's what the questioner is on about. And so I have two responses to that. Number one is there's a long precedent in the literature of researchers arguing in a circle. It's called circular reasoning, and it goes something like this. Number one, we find collagen in these fossils. Number two, this is the second premise. These fossils were deposited 70 million years ago, or insert evolutionary age number. So we find the fossils, they're 70 million years old. Therefore, conclusion, the proteins in those fossils must also be have persisted for 70 million years, period, end of story. Of course, that kind of arguing in a circle, it presumes the very conclusion that it makes. And that's quite common. And that's in the literature, it's in popular articles as well, and we kind of tend to do that too. So it's helpful for us to make sure that we're not thinking in circles, right? Well, here's what I would insert into that premise one, premise two, and conclusion argument. I would insert a third premise, and the third premise says, but the decay rates disprove the possibility or preclude the possibility that proteins can last that long. And so that's what we're faced with. So when I hear one of my colleagues propose a possible solution to their dilemma, I don't have the dilemma, because in the young Earth, like recent creation, recent flood model, those proteins were deposited thousands, not millions of years ago. So therefore, it's expected that some of them might persist. And lo and behold, there they are. But in order to preserve the deep time, the tens of millions of years, we have these proposed scenarios, like maybe it was a bunch of extra minerals, or maybe it was a bunch of extra iron that stuck onto the tissues, or there's one called what I call the toast model, where the tissues got sort of baked enough to where the outer layer of the tissue turned into a resistant layer. All these proposals, guess what? None of them have been tested. They're all stories. So until we get someone to test them, I feel no compulsion to go with them, because here's, if I say that that collagen can last, like Dr. Biddle showed, 10 times longer than we know it can last based on decay rate studies, then I'm actually going against the second law of thermodynamics that says that systems decay over time, and they get less and less organized over time. So the burden of proof lies on the person who puts forth a model to say, hey, these proteins could have lasted much longer than chemistry says they can last. Well, the burden of proof lies on you to say how, and show me that it does. Do the decay rate experiment. None of them have ever done that experiment. So I'm going to stick with the science, which actually points to the Bible, which suggests that these are all fresh looking.
Well said. Great answer. I like the way you put it, Dr. Thomas. The young Earth creationists like us, we're not the one with the dilemma. The fact that we find these incredible preservations is testimony to a young Earth, and it is contradictory to the billions of years paradigm. And so very good. Let's get one more question based on your presentation, Dr. Thomas. This comes in from N9 Nate. Thank you for the question. For Dr. Thomas, can collagen last over millions of years in extremely perfect, stable preservation conditions? And would there be such conditions if millions of years existed?
Well, as to the first question, I feel like I answered that in the presentation. If you'll watch it again, you'll see that I quoted from secular scientists, and they said that collagen cannot last any longer than 900,000 years in theory, based on the measured decay rate, under ideal conditions. So that's extremely perfect, stable preservation conditions, which means no bacteria, no radiation, um, you know, those kinds of things. So the only way to get collagen to last over millions of years that I can think of is to get it so cold, like close to 0 degrees Kelvin, so that the chemistry goes so slowly. But of course, we can't even get it down to liquid nitrogen levels on Earth's surface. So that's totally irrational. Which leads me to the second question. Would there be such conditions if millions of years existed? Well, the more time you give a system, the more opportunities you give that system, especially a system of chemicals, to undergo chemistry, which happens relentlessly. So the more time you give it, the more chemistry happens, and the more opportunities you give that chemical, i.e. collagen or any other biochemical, to undergo chemistry and then turn it into dust, as it were.
Isn't that incredible? 900,000 years in perfect, extremely stable conditions. And even that is far less than the millions of years that they would need to explain. Sam, you said you had a quick follow-up, brother?
Yeah. So I'm wondering, perhaps a critic might respond that it could have been contaminated with something that would preserve it in some way. So on its own terms, yeah, we understand that these things would decay. But is it possible that something like iron or something was contaminated into the biological material, which helped the preservation process?
Well, I suppose it's theoretically possible, but so the burden of proof lies on the person who's proposing that, right? Like what is it that could contaminate this? Something that's available when the creature's deposited. And what contaminant could actually do preservation? And we don't know of any. Maybe there was a giant bucket of formaldehyde that covered the earth and helped preserve these things. But if that were to have happened, even that, what does formaldehyde do? It's called cross-linking. So you have a protein here, and it does extra chemistry to bind those proteins together and turn them into no longer a protein, but some sort of a jumbled mass of polymer, okay, which might last longer. But guess what? We have evidence. We're not finding polymerized proteins as often as we're finding the original proteins themselves and even sequencing them. So you can have, you can have a dump truck or a cement mixer the size of the moon filled with formaldehyde, dump it on the earth over eons and do all this, cross-linking. But what would you end up with? Well, you'd end up with cross-linked biomaterials, but that's not what we find. We find a little bit of that, not due to formaldehyde, but due to heat, basically. But what we actually sequence, what we actually detect, what we actually find is the original proteins, fragments of them, bits of them that we would expect to still remain if they were there for only thousands of years.
Wow, that's awesome. Okay, I love it, Dr. Thomas. I really appreciate those responses. Again, it is an amazing time to be a biblical creationist. Great stuff. So, okay, we got a couple questions now for Dr. G. Dr. G, thank you as well for your excellent presentation on a very important topic. And so I'll bring in the first question, and I can provide some context as well, as this is a common one. Does CET, continuous environmental tracking, explain post-flood speciation or variation? So in other words, Dr. G, critics of the young earth creation position, they will oftentimes assert that we believe in hyper evolution or hyper speciation because they explain all the variations in species in the world through evolutionary processes, right? Natural selection, mutations, so on and so forth. And so I'm curious what your thoughts are on that, Dr. G, and if your model has some good explanatory power on explaining that. Oh, actually, just one sec. Let me make sure your audio. Okay, Dr. G, you're good.
Yeah, the answer is, of course it does, because the main criticism that you just mentioned is the word hyper. They believe that we would have to explain rapid changes in just a very short period of time. So, and following in the tradition of Dr. Thomas, I'll have two parts to this answer right here. First is a lot of what they're talking about in terms of evolution probably really isn't evolution during that period of time. In other words, evolving from a creature like Pakicetus to regular seagoing whales really didn't happen. So there's no need to have to explain those kinds of scenarios with the CET model. But in terms of the diversification that you see, particularly as illustrated by Dr. Werner's skulls, he showed you all of those skulls by the dogs, for example. Many of those changes can be explained by mechanisms that are not just pure genetic mechanisms. As I mentioned in my slide, there are many different ways in which organisms vary and adapt, and not just pure changes to the DNA. And some of those changes that are in those dog skulls are due to epigenetic changes, and others are due to changes in terms of just tandem repeats and other factors in the genome without actually changing the genes for the coding proteins as well. So since CET encompasses many, many different mechanisms, genetic mechanisms, epigenetic mechanisms, other types of non-genetic but heritable changes, and so on, it's quite robust in explaining post-flood speciation and variation.
So is there a lot of morphological adaptability built in to each kind that can come about rapidly, as you've been putting forth, due to environmental changes after the flood?
When you say a lot of course, that means within specific parameters. And right. Some people will confuse the term continuous environmental tracking, the word continuous, which is a time-related term relating to the fact that they are doing it all the time, with an extent expression that it's implying that there's unlimited amount of change, but that's not true. It's an engineering-based approach, so everything operates within specific parameters, including variability and adaptation. So, yes, there is a lot of potential, which probably hasn't even been completely expressed yet, in these creatures as they detect different environmental conditions. And because there is a capacity built in up front, they're able to express different traits towards that. And the capacity is not strictly genetic capacity. And everybody fixates on the genes, but the capacity that the Lord has built in is more than just changing specific genes. It's changing expression of the genes through epigenetic mechanisms. It's changing behavioral things. There's many, many different ways. I published a paper in the 2017 International Conference on Creationism, myself and Phil Gaskell, where in the appendix it lists 22 different mechanisms that are in the literature for expressing these type of changes, including somatic to germline changes, where cells in your body tissue, which are called somatic cells, are actually capable of transmitting information into developing germ cells, which are your sperm and egg cells. So there are many different mechanisms to express this capacity.
That's awesome. I appreciate that response to a very common challenge, Dr. G, and I think clearly we have a great answer to it. And so here's the second one, another common one put forth by proponents of evolution. How does the CET model explain arguments from junk DNA, but specifically dealing with shared pseudogenes, aka genetic mistakes? A little more context, the evolutionists will say, well, we share these mutations or these genetic mistakes with the chimpanzee, for example, and therefore we must have inherited those from a common ancestor in the past.
Right. Well, I'm glad we're finally, actually, just as a preface, getting some questions on biology, because in many respects, in terms of creation evolution, biology is really the name of the game. Yeah. And for so long, we've focused on geology, because in some ways that's the low-hanging fruit. And it's dealing with the age of the earth, and the geology, in some ways, dealing with the layers is is not the difficult challenge that we have to take on, which is Darwin's theory, which was to explain the design of organisms. And it's really the design of organisms which is general revelation, indicating that there really is a creator. And two, it's the incredible design of organisms which shows the wisdom and the power and the genius of the Lord Jesus Christ and expresses his glory. And that's why creationists should really, really be concerned with the biological arguments, because that's really what Darwin is after in attacking and pointing out there is no creator. And so we really need to be getting down to developing theories which explain these features that we find in ways that glorify the Lord. And one of the presuppositions that evolutionists have is this concept of junk DNA, or there are mistakes. And I don't know why anybody would continue to push this, because on nearly every website by every creationist ministry, there are literally dozens of articles dealing with the whole idea of junk DNA, which is rapidly falling apart. The idea that there are all of these mistakes built into the genome, when in reality, it was just ignorance of people that didn't understand what this information was all about. And now, most evolutionists, I can't say in a number, are completely abandoning, at least in terms of their literature, the whole concept of junk DNA. And one of the evidence of junk DNA were pseudogenes. In other words, these were genes which were considered to be functional at one time and were broken and lost their function, and somehow got repeated due to copying from one common ancestor to another, as you mentioned, between us and chimpanzees. But just like most of the arguments for junk DNA, the pseudogene argument is also falling apart as we find function for one so-called pseudogene after another. And we have multiple articles on our webpage at icr.org about pseudogenes, and there were large articles published in the Creation Research Society Quarterly about pseudogenes as well. And so I don't stipulate to the premise that these are genetic mistakes at all. We find function for many of these, and what I would suggest is within an engineering-based framework where we assume that these things have function up front, that we continue to research for those functions of these so-called pseudogenes as well. It doesn't necessarily relate specifically to the CET model, but in terms of what we should be looking for as creationists.
That's a great answer, Dr. G. I appreciate you engaging those questions. And okay, let's get a couple of flood-related questions for Dr. Dan Biddle, who focused on some of the best lines of evidence for a worldwide flood. And here's one, comes in the form of a super chat. So I do appreciate the support and engagement. We've had a lot of positive feedback, gentlemen, from our live chat tonight. Pseudonym, $10 super chat. Appreciate the support. He's wondering about dino fuel. While humans and dinosaurs live together, he says, according to the young Earth creation model, his question therefore is this: how can you believe only dinosaurs alone make up our gas or petroleum, where people that young Earth creationists say live together? Dr. Dan, do you have any thoughts on that?
Yeah, the first observation is that there was at least 3 times, most creationists would say even more, but at least 3 times the amount of vegetation before the flood than we have now currently on Earth. And there's probably multiple factors more than that, because if you look at the coal seams and the oil deposits, they're just vast amounts of this stuff all over the place, and it's on every continent. So we've also learned that coal and oil can be formed rapidly under the right conditions. So you don't need necessarily a time, heat, and pressure. You can just have heat and pressure and a little bit of time to create both of those substances. So where does oil come from? I'm not an oil chemist, but I think it is very interesting when you look at the 2 matching slides of those continents that I talked about. They looked at 12 different types of oil fields that were found with the oil chemistries, and they were matching, proving that they came from the same types of flora and fauna that was rapidly buried. So we do believe that man and dinosaurs lived together. Humans had 150 days to go run for high ground, so there probably not a lot of them lived, but a lot of the that whole biology, all the biology and all the vegetation before the flood was rapidly entombed, sandwiched under a lot of sediment and a lot of weight, and then you don't need a lot of time. You just need heat and pressure to form those things. So hopefully that's an answer for what he was asking at.
I appreciate that, Dan. A common argument put forth by those in the old Earth creationist camp is that there's just far too much coal, far too much oil, natural gas for us to explain in just 4,500 years since the flood. But if what you're saying is true, that let's say 95% of the pre-flood world was green, it was filled with life, then is that really a valid challenge in your opinion, Dan?
I certainly don't think so. I know Dr. Snelling with Answers in Genesis has done exhaustive studies on looking at the amount of pre-flood vegetation, and it's really uncanny because if you look at a coal seam, you think, oh, it's just a bunch of black-looking dirt stuff that's compressed. But if you look at the ratios of green, fresh foliage and other types of products that have to be pushed down in this organic pancake and then pushed down and compressed, it's really amazing to think what that pre-flood world must have looked like. And it doesn't need all three of those things. It doesn't need heat, time, and pressure. You just need pressure and heat, and not a lot of time. And I'm sure the rest of us on this call would have even more exhaustive answers, but that's my perspective.
It's a great answer. Sam, did you have a follow-up?
Oh, no, not right now.
Okay. Appreciate it. Dan, the next question is another common one here, but it's an important one in light of what we are upholding here tonight, gentlemen. The truth of biblical creation, specifically young earth creation. There are many in the world of Christianity. They would argue that the flood, rather than being a worldwide flood, here's the question up here, they'll say that it was a flood simply on the world of the ungodly. And so wherever humans were at that point in time, they'll specifically look to 2 Peter 2:5, and spared not the old world, but saved Noah, the eighth person, a preacher of righteousness, bringing in the flood upon the world of the ungodly. And so, in other words, they don't believe that the scripture demands a worldwide flood, just as long as that part of the globe was flooded where people were.
Well, they can have those perspectives, but they're not talking about Mabul, the biblical flood, the huge, catastrophic, all-encompassing biblical flood, where 60 times in those three chapters in Genesis 6 through 9, we have words like all, everything, everything that sucks air through its nostrils is going to die. And then it's interesting where God says, and I agree and believe that this actually backs up catastrophic plate tectonics. He said, I will destroy them, wicked humanity, with the earth. I'm going to use the earth as a tool to get rid of these people, to scrub them. And so it was not a judgment upon some wicked little tribe or wicked little group. God brought an entire judgment, and in his mercy and in his judgment, those two things at the same time, decided to push the reset button and flushed out everything, because he said, look, every thought of man is evil continually. And that's what moved God to the place out of mercy and out of judgment to say the whole thing is gone. I'm going to send Mabul, the Hebrew word for the big monster flood. And he did an amazing job. We reviewed it tonight for basically for 2 hours. You look at the 1 hour of sediment that Dr. Werner talked about in the Grand Canyon, and you look at the Sloss mega sequences that Brian talked about. There's no room for little flood, big flood.
Amen. You guys all did an excellent job showing exactly why science confirms the Bible. So that's a great answer, Dr. Dan Biddle. Appreciate it. Next is a question for Dr. Carl Werner. And so a question we have here comes in from Apologetics 101. There's a lot of discussion regarding castoricata. Hopefully I said that right. What is your response to the accusations by the critic regarding this living fossil? Is that something you're familiar with, Dr. Werner?
Yeah, that was a animal that was found in the Jurassic layer, and it appeared on the cover of Science, and it looked like a beaver, and they called it a Jurassic beaver. But I have never seen the bones, and I've never seen a recreated model of the bones at a museum. And basically, someone needs to go out and do a side-by-side comparison with the modern beaver and modern beaver and this Jurassic mammal to see if it is in fact closely looking like a beaver or if it's just a totally unrelated animal that looks alike. So that would be a project for somebody to do that side-by-side comparison, because you cannot trust the evolutionists just because they put a different genus and species name to say that they're different. You know, someone have to go look and do the work, but I have not done that.
Appreciate it, Dr. Werner. This next one, in many ways, was addressed tonight, especially in your presentation, Dr. Werner. But anybody on the panel, if they'd like to add some thoughts, feel free to do so as well, since it is a major one that's been issued towards us young Earth creationists for years. How is the fossil record sorted in an order convenient for evolution? Right off the bat, Dr. Werner, we're going to hand it to you. But as you demonstrated, we find living fossils, we find patterns in the fossil record that don't seem to be convenient for common descent, which is what you presented. But Dr. Werner, if you'd like to elaborate, go ahead.
I'm not sure that they're sorted, but they're assembled onto a piece of paper as a geological column. So if you believed in evolution, you'd have to say it started with a bacteria, then it went to a small invertebrate, and then went to so if let's just say creation is true and evolution was not, but you want to demonstrate it, you would select the first layer to be the lowest layer, to the ones that have just the microscopic bacteria and very small invertebrates. You could not select for the lowest layer one that had a dinosaur in it. And believe it or not, the dinosaurs aren't in the middle layer, because sometimes you find them at the top layer where you go to the dig sites, and sometimes they would be on the bottom layer next to the basement rock. So you have the capacity, and I have not checked this out. I wanted to do the interviews, but I haven't done those. You have the capacity to assemble a geologic column and make it look real because it's on paper. But why do you select that layer to be the lowest? Because there's a lot of different layers that are right up against the basement rock, but you have selected this one. And even like the lowest layers, like at Ediacaran and Flinders Range in Australia, or Burgess Shale, those lowest layers, they're not the lowest layer, and they're not laying next to the basement granites. They're a layer, and there's a whole bunch of layers above them and below them. So I'm not sure that it's real. I stand to be corrected, and I would hope to do interviews with a geologist about that. But there's many other problems that I would have to explain why I'm doubting this geologic column, but that would be one. I'm not sure that they are assembled that way.
Thank you, Dr. Werner. Anybody else on the panel have any thoughts or anything they wanted to add to that specific challenge, the ordering in the fossil record?
Brian's dying to say something.
I'm happy to, yeah. So according to the evolutionary trees that we keep getting shown, you have, like Dr. Werner was saying, here's my evolutionary tree, okay? You've got bacteria, and then plants, and then this evolves, and then you should have this burgeoning, flourishing new. You have to have vertebrates, come from invertebrates. You have to have plants come from the same ancestor as all the animals. So you got to have hundreds and hundreds of different, completely different body plans, phyla, even. And so these are supposed to emerge later in upper layers, okay? But what does the fossil record show? It shows the evolution. This evolutionary plan, this evolutionary tree, is actually inverted, and we have all the phyla, including vertebrates, are already present in the Cambrian, in the very first layer. It's called the Cambrian explosion. And so I completely disagree with anyone who would say that, or with the premise that lies behind this question, that the fossil record is sorted in an order convenient for evolution, it's actually sorted in an order that's exactly the opposite, or very inconvenient for evolutionary trees. It's an inverted tree when we look at the fossils.
That's a really good point. Just this sudden appearance of jellyfish, coral, and all these massively different kinds of organisms, this Cambrian explosion, very inconvenient for evolution, not convenient. Dr. G, did you have any thoughts?
Well, I think Dr. Thomas really nailed it. It's not sorted. I would totally disagree as well. That's convenient for evolution. It's completely upside down. In the lowest layers where you find major numbers of fossils, you're finding the major body plans already showing up there, and there is no evolutionary ancestors to them in the fossil record whatsoever. So this isn't convenient for evolutionary thinking whatsoever. In addition to the fact that you find, as Dr. Werner already mentioned out, you find marine fossils in every single layer. And not only do you find them in every single layer, they are the majority of fossils in every single layer. So what I really find in the geologic record is evidence of a worldwide flood in abundance.
Amen.
Yes.
Well said. Dr. Biddle, and anything you wanted to add to that question?
I think for me, I find them conveniently sorted in the order of the flood. I've looked at the fossil evidence for years, and for me, what I find is that the seas, the trilobites and those creatures, they bought it first. They're covered with mud and tens or hundreds of feet of it sometimes, and then you go up from that, you're going to have the shallow seas. That's exactly what we find next. Then you go up, you're going to find the lowlands, and that's what we find next, and then layer after layer after layer. And, in California, you have messy geology. You've got Jurassic stuff on top of Cretaceous stuff. So it's not as clean as people would suspect. The only place you're going to find that geological column is in a textbook. It's not nicely sorted, as they say. The flood created a big geological mess, but the fossils generally are in an order that's going to show their burial order with respect to things like body mass and mobility. That's exactly what we see in the fossil record.
Fantastic answers all around, gentlemen. We've got time for one final question for the night. It's been a great night of presentations and answers here in our audience Q&A period. We can end it with a bang in a similar manner as to how the evolutionists argue or assert that the fossils are ordered in a way that's convenient for common descent. You gentlemen prove that assertion wrong. They'll also argue that the layers are ordered in a pattern that fits not only billions of years, but common descent. And so they'll say, well, if most layers were laid down in a year, why do lower rocks date older than higher rocks? And we can consider this final question, gentlemen, just a general question for anybody to answer and anybody to offer some input.
I'll do it. So the way you get everything in order, where the lowest layers are the oldest number of years, and you come up, is that you test that layer that you think is oldest, and you test it and retest it and retest it with multiple different methodologies. And once you get an answer you like, then that's your number. There was a fossil in human evolution called 1470 that was tested 200 or 300 times, and the evolution scientists dated the fossil 200 million years. It was only supposed to be 1.7 million years old. It was 200 million years, 180 million years, 150 million years, 30 million years, 20 million years. You just keep testing it until you get a method and a result you like, and then you say that's what it is. There is no, those dates don't work like that, where you just test one rock and, that's the number. No, because each scientist then comes up with a different number. So I totally disregard any dating, because it's always non-repeatable, which is a basis of science.
Very good, Dr. Werner. Dr. Thomas, it makes me think of your initial presentation that kicked off our biblical creation seminar tonight. The collagen, the proteins, the red blood cells, all of these things preserved that we find, doesn't that in a way indirectly refute what they're saying about the geological rock strata and the ages they give?
Well, when I'm looking at collagen or proteins, um, let me just say this about it in general, real quick. Science cannot assign a date to anything. We don't. Science is not the tool to use to determine how old anything is. The only way to use science to determine the age of something is if you had a time machine and could could measure whatever it is, whatever decay process you're measuring at the moment that the decay started. But it does give us clues. It does give us ranges, and so that's what I think collagen gives us. So if I have collagen in a fossil that's in the Cambrian, assigned 500 million years, and it's got collagen in that fossil, and then I see collagen as well, or a protein that's in the uppermost rock layers, it really is a tight match to the expectation that all these layers were deposited in the same year or within a similar time frame not that long ago, which is exactly what we'd expect if the Bible's true. So I think perhaps this questioner is asking something more detailed, and I would first of all, the layers they're sedimentary layers, and so you can't directly date those either. But it's the igneous rock layers like granites and such that we can use isotope systems to assign ages. And like Dr. Werner was saying, let me just throw this out there. There's a basalt layer that's beneath the layers of the Grand Canyon that's supposed to be over 1 billion years old, and then there's more basalt that erupted after the Grand Canyon, after the layers were deposited and the canyon was carved, and then an eruption happened and the lava fell down into the canyon. And so we dated that lava, and it actually dated at an age, using the conventional isotope systems, as older than the basalt that was beneath all those layers.
Wow.
So it effectively erased all those imaginary years. So I agree in principle with Dr. Werner's distrust in the isotope systems, but there are patterns in those isotope systems. I think it's beyond the discussion for tonight, but you can investigate those using the ICR rate project, and it's all online for free, icr.org/rate, if you want to go read for the next year and find out more about that.
There's a lot of material there. We actually just did a seminar on CPT with Dr. John Baumgartner. We went about 4 hours and we discussed a lot the ICR rate project and accelerated decay as a way to answer this question. So Dr. G, Dr. Biddle, anything you gentlemen wanted to add to this question?
I do agree that there are. Oh, sorry. Go ahead, Dr. Okay,
no, I wasn't going to add anything. I thought Dr. Thomas and Dr. Werner did a great job, so nothing for me.
I agree. Great job. It's the ash layers that they date, and you do find that you can find relative differences, but cranking them from the radiometric decay to an observation with a human meaning like years is an interpretation on the data. And personally, I've been on an international committee of carbon-14 dating experts for years, and these are the guys that actually pull the handle on AMS machines. I mean, this is carbon-14 dating at the highest level possible, and none of us are afraid of the outputs of what they're showing about the age of the Earth. And it's because the deeper you go back in time, the more assumptions that you have to take into consideration when you interpret your findings. And if you look at carbon dating alone, things like volcanic ash and forest fires and solar flares and Earth's magnetic pull to the moon, all kinds of things can really change carbon-14 dating. And if that's true for just carbon-14 dating, what about all the other methods that are trying to infer dates of hundreds of millions of years? So it's actually more scientific to have higher levels of scrutiny about those outputs, and especially when you go and look and try to validate the dates that they're coming up with. When you look at the Mount St. Helens rocks, for example, rocks we watched get form in the 80s and 90s, and you take them into a lab, and those rocks that are only decades old are returning dates between 280,000 years and 3 million years old, all for a rock that we know is just a couple decades old. And that's not just one example. There's lots of examples like that. So relative dating is okay with me, but absolute dates, when you crank it back to years, requires way too much faith for me.
That's right.
Would it be fair? Would it be fair to say that the observable biological decay rates found throughout those stratigraphic layers contradict the dates made by certain dating methods of the layers themselves?
Well, what would you say, Brian? Because I know that you found collagen in the Triassic, Jurassic, and Cretaceous through all kinds of layers, right?
It would seem to present a contradiction. If we take the conventional age assignments to those layers, right, and then we said, well, if this is 70 million years old, then why does it still have protein? That's definitely a dilemma. I think biblical creation solves it. But what you have to do at that point is you have to say, well, what's going on with these radioisotope age assignments? Let's say you do have, like you said, Dr. Dan, an ash layer that's in between sedimentary layers, and you can date the ash layer or date it. Well, then you run into what Dr. Werner was saying, which is we just keep running tests until we get the number that agrees with the chart that's in the technical books that shows this chart of evolution. And it turns out that the chart that shows when things happen in terms of evolutionary time, that is what reigns supreme, not the actual data. We have to, what they do is they fit the data, they fit the data into that evolutionary grid. So, but to your question, Sam, I don't think that I'm not assigning a collagen age to a rock. I'm just saying that there's enough information, age-related information, to allow us to discern between these two competing models. They're vastly different. The evolutionary model says millions and billions, and the Bible says thousands. And what that does is, once we see these proteins and many other evidences that point to recent creation, then that you take this whole evolutionary timeline and just shrink it down like an accordion, and then boom, you've got me. I came from Adam. Adam came from God, and there I am, face to face with God. So I kind of want to leave our audience thinking about God himself. I mean, why not? We have to meet him one day, or we get to meet him one day. If we have to meet him, it's because our sins are still before us. But if we get to meet him, it's because we have trusted in his son, the Lord Jesus, to rescue us from the penalty of our sins. And may that be the case with all of our listeners.
Wow. Amen, brother, Dr. Thomas. That is an excellent way to wrap up tonight's biblical creation seminar, which I have been extremely excited for. I know you have as well, Sam, and many in the audience. And you gentlemen did not let us down. 4 excellent presentations and a really thought-provoking, engaging audience Q&A, where these questions and the feedback that comes in gives us the opportunity to elaborate a lot on the topic and what has been said throughout the program. So Dr. Thomas, Dr. G, Dr. Biddle, and Dr. Werner, I really appreciate the time that you've given to us. I know how busy you all are for tonight's Science Confirms the Bible seminar. And I do want to encourage anybody in the audience who has been edified by tonight's content and seminar, do check the description box of this video where you can find links to ICR. You can find links to Dr. Carl Werner's work, and also Dr. Dan Biddle at Genesis Apologetics. And so with that, gentlemen, thank you so much for doing this.
Thanks for having us.
Thanks, Donnie
Lots
and
of fun.
Sam.